to give me this chance to share our some recent progress. So today the title of my talk is revealing the cell heterogeneity, development trajectory, and the non-subpopulations with the immune function in megakaryocyte. So as you may know, actually human pluripotent stem cell, including iPS cell or embryonic stem cell, have been widely used as a potential source for dividing all kinds of blood cell, such as this lymphocyte, also red blood cell, or megakaryocyte and platelet. So in my lab, actually in the last couple of years, we have keep using a co-culture system. What we have done, we culture this human pluripotent stem cell with this mouse AGMS3 stromal cell. After this co-culture for 5 to 7 days, you can see this beautiful hematoprogenitor cell can grow up from this sac structure. So this sac actually grew in suspension, so it's very easy to observe. So when we conduct some immunostaining, we can see this hematoprogenitor cell can be aligned in a beautiful cluster. They are 45 positive, and they can line beautiful in this sac structure. And actually, this sac structure is formed spontaneously during this co-culture system. Also, if we put this cell into the co-culture system, you can find this can form all kind of different colony. This means they can really have the potential to form different kind of blood cell. So our interest is try to induce this hematoprogenitor cell to megakaryocyte. As I show you here, actually these are clearly they show larger size. Also this platelet, you can see here, this kind of fiber structure. So here's another video to show how this megakaryocyte can form the proplatelet. So here show there, this big megakaryocyte. They can, after the culture system, they can gradually show this kind of fiber-like structure. This is a proplatelet. So later on, they will form the platelet. So by using this system, actually, we have been keeping study this mechanism about this megakaryocytes, also thrombopoiesis. So as I show you here, and this can, in wild types, you can really form this proplatelet beautifully. However, if I remove this one gene called MEIS1, MEIS1, by gene knockout, you can see here, you can only find very few megakaryocytes can formed. Also, you can never find any proplatelet can formed from this co-culture system. So this actually, we have been keep using these systems for our study. So in our study, actually, we have raised some interesting question. Why some MK never produce any planet? So it's actually in our co-culture system, because our long-term purpose is to try to get a large amount of planet for the transfusion. However, in our experiment, we found actually in many times, actually some megakaryocytes, they never can form any planet. Also, we wonder in this MK, they should have the equal potential, or they are just actually they are biased. So to answer this question, to come to the two part of the part one of my talk. So we call it decoding human megakaryocyte development. So this part I mainly focus on the early development stage. So I have to point out this work have been collaborated with Dr. Bin Liu's lab and also Dr. Li Hongshi's lab. So this part of work have been accepted by Cell Stem Cells. This should be online within 2 or 3 weeks. So what we have done, actually, we isolated this blood cell from yolk sac, also the fetal liver, and then put them into a 10x genomics for the single cell sequencing. As you can see here, actually, from both yolk sac and the fetal liver, we can identify a very reliable MK subpopulation in from both sides. Certainly, you can get a lot of other cell types. Also, we look at the morphology of birth cell, as you're showing here, actually very interesting. From this megakaryocyte from yolk sac, you can see actually this shows 2 different morphology. One is actually very small, and also certainly they are CD41 and CD42 positive. But you can see here, a lot of cell types, this is some very small, beautiful protrusion. Actually, we wonder they might be there's some proplatelet. Actually, how they form the proplatelets, they might be different with this megakaryocyte from adult. So this, I guess this is the first time we showed some beautiful structure here. Also from the fetal liver, there also show some different morphology about the megakaryocyte, but certainly they are more just like the megakaryocyte from bone marrow. So they are polyploid. Also, there are some like a propellant structure have been formed around the megakaryocyte. We have conducted some ploid assay about this cell. As you can see from here, actually in the yolk sac, very few megakaryocytes actually is polyploid. But however, in the fetal liver, you can see clearly more megakaryocytes have been goes to the polyploid stage. So in the very beginning, we just simply put this megakaryocyte from the fetal liver and the yolk sac, and then conduct the assay and compare them. We found something very interesting difference between them. Actually, as you can see here, actually we found some glycase gene have been enriched in yolk sac. In contrast, actually there's not a gene enriched cell cycle, or they have been enriched in fetal liver. So this actually, I guess, they are maybe correlated with their megakaryocyte from yolk sac. From fetal liver, actually they have a higher proliferation potential. We also looked at the cell heterogeneity about this megakaryocyte from these two sites. They have been divided into 6 different subpopulations. Actually, we found some very interesting subpopulation here. The one is called a cerebral palsy subpopulation. They actually express a lot of genes involved in megakaryocyte development and platelets in contrast to other subpopulations. So this actually partially explained our previous result why some megakaryocytes actually they have less potential to generate the platelet. That means this actually among this the megakaryocyte population, we do have some special population they are biased to produce this platelet. Another subpopulation very interesting we identified is called a niche support subpopulation. We found actually they clearly enrich some lot of gene. Maybe some of them are familiar with it. Actually, they are benefited this HSC development or other cell type. We also identify a very interesting subpopulation called the immune subpopulation. We have named as the immune subpopulation. Actually, you can see here, actually they express a lot of gene, very specifically enrich this immune associated response. I will show you more data later. After this 10x genomic sequence, I also confirmed this cell heterogeneity with the SMART-SIG2. We isolated more cells from the yolk sac and the fetal liver, and I confirmed this the cell heterogeneity. They are pretty much the same thing. Also, we did the trajectory assay for this cell. Actually, interesting, we found some 2 different directions. In direction 1, actually, they enrich a lot of gene, seemingly more likely close with this classic function of megakaryocyte or planet, like hemostasis, also the planet deregulation, and some other function. However, there are actually clear there are another direction. They're enriching a lot of megakaryocyte with this cell substrate adherent and nucleoside differentiation or microphage migration. So we wonder actually this direction actually may generate more megakaryocyte to associate with the immune function. So just quick summary about what we have found so far. Actually, we identify a comprehensive transcript landscape of this MK from human yolk sac and fetal liver insects in a single cell solution for the first time. We also found actually this yolk sac and fetal megakaryocyte from yolk sac and fetal liver show some interesting difference between the cell cycle and metabolic genes, also morphology and cell ploid. So we found this actually they're quite heterogeneous, and also this they have simply have 2 different trajectory for this to generate this megakaryocyte from this embryonic stage. So after we get this data from in vivo, we wonder what happened in vitro. So we have taken advantage of the system I introduced in the very beginning. Actually, we use this co-culture system. We take the cell from this 12 days after co-culture or 6 more days for the megakaryocyte differentiation. So you can see from here, actually, from both time points, we identify the subpopulation of megakaryocyte from both time points. So we just take this, we call it day 6 megakaryocyte differentiation. We found actually this, indeed, they show also the megakaryocyte show the cell heterogeneity. But for this population, we can only identify 5 different megakaryocytes. But indeed, we found actually there interesting subpopulation enrich a lot of innate immune response gene, also this microphage activation, as we show in the in vivo. So look like this subpopulation megakaryocyte actually they do exist. It's not an artificial one. we take a better look about this gene. We compare this gene enriched in MK4 with other MKs. Clearly, we say this is a microphage activation or like activation or innate response gene have been enriched in MK4. They express, they have considered like a 5 to 10% in vivo, but in vitro, they simply they have just less than 5%. But every time we found actually very reliable population about this we call it immune subpopulation. So besides this cell heterogeneity, we also have interest about to find some interesting marker to track this human megakaryocytes, because actually they don't have too many markers to track this process so far. What we have done actually, we did the PF4 correlation assay for this megakaryocyte from both model, either from jiaxi, also this human-derived endothelial cell. Interesting, we found identify one marker called THBS1. This actually showed a very beautiful expression correlated with the PF4, as you show from here, or this in vivo one. So to track this, when this is their function, actually we have tried to establish the THBS1 GLP reporter. What we have done, actually put this GLP gene in the THBS1 nucleus, and then establish this THBS1 reporter cell line, then induce to them to megakaryocyte or platelet differentiation. Interestingly, we found actually this GLP can successfully mark this megakaryocyte, also the platelet. That means this marker, this THBS reporter indeed works. So then we try this dynamic expression during this whole different process. First, we look at we compare the expression with this CD42, a reliable marker for the megakaryocyte. Interestingly, we found actually much earlier than the expression of CD42. You can find actually in the very early stage, the THBS1 GLP have been started to express. Also, we did a flow cytometry assay. We found actually, as you can see here, actually in the very early stage, there are a lot of CD31 positive endothelial cell. They already have some THBS1 GLP expression, because this is actually a very unexpected result. As you know, actually, we should get those hematopoietic progenitor cell stage, but not endothelial stage. This can express this marker. So to further study this, we have conducted some function assay. We isolated this GLP positive. That means this they already have the THBS1 expression and the CD31 positive endothelial cell. Then compare the different potential. Very interesting. We found that the GLP positive endothelial cell already have this much higher potency for this megakaryocyte differentiation. Also, they can generate more platelets than this GLP negative endothelial cell. That means actually in the earlier this endothelial stage, they already have this featured bias. That means they already have this bias potential to the megakaryocyte differentiation. This is a very unexpected result. We also further validated this by isolating this GLP positive or GLP negative CD43 positive hematopoietic progenitor cell. Because this progenitor cell, actually, this is, as I mentioned, is a hematopoietic progenitor cell. So certainly they have also the difference. That means this THBS positive hematopoietic progenitor indeed have the higher potency for the megakaryocyte differentiation or planet. So a quick summary about what we have done. And we have found actually human embryonic stem cell derived megakaryocytes actually partially recapture this in vivo MK heterogeneity. Also, we identified a very interesting marker called THBS1 as an early marker for the MK bias embryonic endothelial stage. We propose actually there might mean this. Certainly, we need more evidence, but we can certainly speculate this megakaryocyte actually may be originated very early stage from this endothelial cell. Certainly, this THBS GLP reporter cell line could be used to trace the megakaryocytes and the thrombopoiesis from embryonic stem cell. So right now, we already established this THBS reporter mice. Hopefully, we can show some exciting data in future. So here is the working model for this part. Actually, we found we have isolated the human megakaryocyte from the yolk sac and the fetal liver, and get the single cell transcription landscape of this megakaryocyte. We prove they are very actually highly heterogeneous, and by using two different directions, actually they can generate this classic, we call it classic megakaryocyte. Also, they have systemically can form an interesting population about the immune MK. And we have some experimental evidence to show, but I didn't show here today. Also, we identify the THBS1. They can really mark this endothelial cell in the very early stage. Even as early as the endothelial stage, this cell can show some bias to the megakaryocyte differentiation. Also, we established, as I mentioned, we established some reporter mice. Hopefully, we can certainly to trace all the different processes of our megakaryocyte development. So now I move to the second part about this my talk, and right now I just show you this in the very early stage. So what happened in the bone marrow? So with the help from Dr. Tianfei Wang's lab, and we have successfully isolated this human megakaryocyte from the human bone marrow. I have to point out actually, because this human megakaryocyte is very big, also very fragile. So it's so far nobody have really reported can successfully isolated this megakaryocyte successfully. So in our study, we have been successfully isolated this. We sorted this CD41, CD42 megakaryocytes, and then we pick them and send them to the SMART6. As I shown here, actually you can see this. The integral megakaryocytes have been isolated successfully. Also, you can see here, actually, they are very big, and they have very high, more than 50% is polyploid megakaryocyte. Actually, in our isolated strategy, actually, we didn't get a lot of like low ploid megakaryocyte. Actually, in contrast, we get a lot of more mature megakaryocyte. Also, we did the heterogeneity analysis. We found actually they have 5 different subpopulations, and they all express the PF4 and the CD41. Also, again, we found actually there are very interesting subpopulations. They enrich a lot of genes associated with like leukocyte activation and the inflammatory response and the leukocyte activation. So as you're showing here, actually this is pretty much similar with the observation in the very early stage, also the in vitro system. So since we have identified this immune MK in multiple systems, so we're wondering what is their function and what is their special. So to do this, we have to identify there are some useful markers. So we have tried to identify some new marker for this immune MK. What we have done actually in the very beginning, we try to identify some more mature marker for this MK, because we wonder so far actually like some CD41 or CD42, most of them actually their expression cross to the MEP or MKP and also as well as this MK stage. So we have tried to look for some marker really just only express megakaryocyte, but not like a MEP or MKP. We think this might be helpful to identify this immune MK. So we have been able to identify a gene called CD148 or PDPRG. Actually, they show the beautiful expression, and we also confirmed the expression in vivo, both in human bone marrow, also this mouse bone marrow. You can see here, actually, they very small megakaryocytes. They never express CD148, but in the more matured megakaryocytes, they do express this CD148. Also, we are looking for some special marker for this immune MK. So what we have done, actually, we try to overlap this surface marker between this immune MK we identified in our 2 different systems, is in vivo, also in vitro. I did overlap. We have been identified some potential candidates like CD48 and CD53. And from the bioinformatics data, you can see also some kind of specific expression. We have picked up CD48 and do the validation. From this expression profile, you can see similarly they are quite very specific. Some other markers do enriched, but we found actually they are not that specific so far. So again, we did the immunostaining to confirm this expression. We found actually CD48 and the expression indeed in both human bone marrow and also the megakaryocyte from mouse bone marrow. So after we find this markers, the next question we try to ask is what is their function, how they generate. So first we take some in vitro model to study this. For the in vitro model to derive this megakaryocyte, we have incubated them with RPS, also some gamma interferon to like modern this immunostimulation to see how they respond. Actually, interestingly, we found that this percentage of CD48 megakaryocytes actually indeed upregulated in a dosage-dependent manner in both systems. You can see here. Actually, that means the generation process of our CD48 immuno-MK, the Zhou can take the response. So what happened in vivo? So we also take advantage of the in vivo model, and we inject the E. coli into the mouse, and then check this dynamic about this CD148, CD48 positive immuno-MK. As you can see from here, actually this CD148 immuno-MK actually they can operate very quickly. They can peak at the 38 hours after this immunostimulation. That means they can really respond in both in vitro and in vivo. As I mentioned, they are peak at the 38 hours. So we just take the very preliminary try to isolate this megakaryocyte after stimulation, 38 hours after the stimulation, and then did the SMART26. And we have been identified in 2 different conditions that MK from 2 different conditions, and like each one had like in total we have almost got the like 850 megakaryocytes. We did the cell heterogeneity assay. They pretty much the same thing with the human bone marrow. So we look at the expression of CD48. Actually, could you see actually in most MK5 actually they clearly show much higher expression about CD48. Also, you can see here they clearly for this MK5 they have more after infection. That means they have been upregulated after this immune response, immunostimulation. So we just keep thinking about what are they doing, and we look at their experiment profile, and we found actually there in this MK5 they have a lot of immune receptor, and they very specifically. You can see here the toll-like receptor and also other receptor. So we have confirmed the expression with the immunostaining, also this flow cytometry assay. We also look at this immune mediator about this MK subpopulation. We can find actually there are a lot of interesting immune mediators have been also highly enriched in this MK5, but not in other cell population. Also confirm the expression by immunostaining, also the flow cytometry. But besides the expression about the immune receptor and the mediator, we wonder what is their function. So we did some in vitro assay first, and many people have reported actually the megakaryocyte can really integrate with this neutrophil. So what we have done actually, we incubated this directly incubated with this megakaryocyte and with this neutrophil. As you can see here, there's some preliminary data show, and this CD48 megakaryocyte actually indeed can accumulate or integrate with them, and they can aggregate more neutrophils than this CD48 negative megakaryocyte. We also did some observation in vivo. As you can see from here, we did some immunostaining. We found actually after infection, clearly see actually there are more neutrophils simply accumulated this around this CD41H megakaryocyte. We had did some quantitative assay, and there's some very significant difference. So here as a summary about this part, and we have conducted this heterogeneity assay about this megakaryocyte from adult bone marrow, and both in mouse and human, and we found actually they are pretty much the same thing. They are quite heterogeneous, and also there are after inflammation, actually this is we call it immuno-MK, actually really responsive. They can express a lot of immune receptor and also immunomediator. So they can so in partner they can really integrate better with this neutrophil. We certainly we need more evidence, and hopefully in future we can really conduct some transplantation assay or other genetic way to get a better interpretation about their function. So this is my work, and the first part of my work have been conducted by Hongtao Wang, the research assistant, also PhD student Xiaoyuan Chen, and Jian He from BNU SNAP, also the PhD student Changlu Xu from Li Hongshi SNAP, also the doctor from Yanghua have helped us a lot for collect the sample. So the second part actually we this conducted by my research assistant from Cui Liu and Dan Wu and Mei Juanxia, two PhD students. As I mentioned, this is the first part of my work is collaborate with Dr. BNU SNAP, also Li Hongshi SNAP. The second part we can get a lot of help from Dr. Tao Chen, and also the Xingao's lab, and Dr. Ernie Jiang and Xiaofan Zhou, also the Qianfei Wang have asked a lot for the collected sample, also the method to isolate the megakaryocyte. So I guess I will stop here and take a question.